Mitosis is the simple duplication of a cell and all its parts. It duplicates its DNA, and the two new cells, daughter cells, have the same pieces and genetic code. Two identical copies come from one original. Beyond the idea that two identical cells are created, there are certain steps in the process. Before a cell divides, it is in resting phase. The cell in resting phase is not actually resting, since it has to maintain its reservoir before it undergoes division. This phase is called interphase. It prepares the cell for division, that is to say, protein synthesis, DNA duplication, and enzymes. Here, we start with prophase. The chromosomes start coiling and become condensed. Nuclear envelope disintegrates. Gene transcription ceases during prophase and does not resume until late anaphase. The nucleolus also disappears during early prophase. Centrosome gives rise to mitotic apparatus, that is to say, generation of spindle tubules. Metaphase. Now all the pieces are aligning themselves for the big split. The DNA lines up along a central axis, and the DNA, chromatin, has now condensed into chromosomes. Two strands of chromosome are connected at the center of the centromere. The tubules connect to the centromere, not the DNA. Anaphase. The separation begins. Half of the chromosomes are pulled to one side of the cell. Half go the other way. When the chromosomes get to the side of the cell, it's time to move on to telophase. Telophase. Now the division is finishing up. This is the time when the nuclear envelope begins to synthesize again along the separated chromosomes. Cytokinesis pinches the cell membrane in the middle. Cell membrane closes in and splits the cell into two. Now we have two separate cells, each with half of the original DNA.